Well, thank you, Professor Gander, for such a great setting. Let me go back. Where's the page? We're going to try to talk a bit about the future of neurosurgery, not next year, but maybe a decade from now, and see how things may change for a number of us. You may remember that the Nobel Prize this year in medicine or physiology was awarded for microRNAs. And in fact, there's over 2,000 of these now have been described. Little history, it was over 130 years, or about 130 years ago that they first transplanted into the brain. And a lot of you will remember Madrazo and colleagues in Mexico for Parkinson's disease about a century later. Stem cells were used for spinal cord injury in particular. And you notice there on the top left, the drop-off after 2016 in a number of trials. And that was because the results were not very successful. But I think hopefully by the end of this talk, you'll see that it's a lot more promising now than it was just a few years ago. The research has continued both in spinal cord injury and TBI, traumatic brain injury, for stem cells. And part of the reason that it's a hopeful field now is it looks like we can combine the stem cells with these microRNAs, again, the Nobel Prize winning. Just quick review of nano. If you look at the upper right, a child's about 1 meter tall, red blood cells about 10 microns. But the big thing about nanotechnology is we're assembling molecule by molecule or atom by atom rather than cutting things smaller as we as neurosurgeons like to do. You're all familiar with these phases of spinal cord injury. First phase, pro-inflammatory, the first couple of weeks, and then the anti-inflammatory, the M1 or M2 phases, splenic macrophages, and then bone marrow macrophages in phase 2. And the microglia have the same pro-inflammatory phase and anti-inflammatory phase. And you can see here, the microglia, like the macrophages, have these M1 bad guys, the pro-inflammatory, the M2 good guys. And here we see some of the effects of the M1 versus the M2s, and at the bottom, all the things we'd like to have happen are facilitated by the M2s and inhibited by the M1s. But the mesenchymal stem cells can help regulate that conversion to the good guys from the bad guys. And one of those means that we're going to elaborate on is the exosomes. If you want to read a review of this topic, this is a nice article in Brain quite recently. Well, you're all familiar with stem cells, but you can see these microRNAs, literally now hundreds of them, can be produced by these various stem cells. Some are good, some are bad for neuroregeneration. And you can also bioengineer, not the stem cells, but the exosomes that the stem cells can secrete. So you can manufacture them with the content that you want to deliver to a certain tissue. We're all familiar with the mRNA vaccine. It's actually a lipid nanoparticle you see in the upper left. If you inject mRNA, in your muscle, it's going to degrade before it gets to the cells. So you have to encapsulate it in a lipid nanoparticle. That's basically an exosome or extracellular vesicle. There's just a little history of the exosomes were described over 40 years ago, but they're basically if you want to call it the FedEx or the DHL of the body. Cellular communication is this is a whole field we really didn't know about until the last couple of decades, of how cells communicate with each other. And you can see here the tenfold increase in publications over the last decade on exosomes. So what are they? You can read their non-replicating lipid bilayer spheroid structure, basically like the mRNA vaccine. And you can see the top I showed in the previous slide, it's a multivesicular body, forms this exosome, and it's targeted to a target cell. In the center you see the content can be proteins, nucleic acids, but one of the most important things is you can use this to deliver whatever you want into the patient. Also, tumors will shed specific exosomes, ischemic conditions, the same thing. And you can also use it for therapy to specific organs in the tissue, in the brain or other tissues. This is a little blow up of the formation and what the exosomes can contain. And we're going to talk particularly about microRNAs just because they've become so important. And this is getting one more level higher in the complication. You can see all the receptors that are on the surface, so you can really target very specifically. And then finally, it gets even more complicated. as we discover more about this, and there's probably going to be thousands of these that will eventually be described. Well, this is from 4 years ago, and you can see exosomes are involved in cardiac repair, wound healing, autoimmunity, tumor development, disc disease, and obviously COVID vaccine. So again, if this was publications from this year, it'd be much more involved. Just one example of how this can work. You can form an exosome with whatever drugs you want to deliver, say, into the brain, inject it, usually intravenously is the most simple. These are so small they cross the blood-brain barrier. I didn't make a point of it. It's about 100 to 150 nanometers in diameter, so it crosses the blood-brain barrier and goes directly into the brain, which the stem cells wouldn't do. And again, these are examples of TBI, of the ways in which these exosomes can affect neural differentiation and inhibits glial formation, for example. Same thing in spinal cord injury. You can say this is going to get more complicated year by year, but I think you just get an awareness of how much work is going on right now and how rapidly the field is changing. Ischemia and hypoxia can really lead to this polarization, but stem cells that secrete certain exosomes can facilitate going from M1 to M2. And this is just an example from 2 years ago of microRNAs involved in ischemic stroke. And you can see there's probably a couple dozen there. It's probably a couple, more like 100 now. And this has hit the popular press with this Scientific American article a few months ago. Well, to show you what these mRNA or microRNAs are, I won't go through the whole steps there, but basically it's formed, and then its effect is really to inhibit or degrade the messenger RNA selectively. And so that's how it tends to affect bodily functions and disease processes. And you can say that 22 nucleotides in length, they're not coding anything. They're called non-coding mRNAs, very small. But they're crucial for normal development, and if they're abnormal, they're associated with many disease processes. And this just shows some of the microRNAs and various aspects of neurodevelopment, synaptic transmission, et cetera. And so I've talked about microRNAs, but there's other non-coding RNAs, the LNC, or long non-coding RNAs in the upper left, and the circular RNAs in the upper right. Probably over the next few years, we're going to find these are a lot more meaningful than we understand right now, but the microRNAs are really have become very influential in our ability to communicate cell to cell. And I just go over what's in red there at the bottom. The punchline is these microRNA chain, they bind to part of the messenger RNA to degrade or inhibit it. So let's look at some clinical applications. Glioblastoma. MicroRNAs can have a positive or a negative effect on all these techniques, the evasion technique, or the ability of glioblastoma cells to avoid natural immunity. It can affect the immortality of the cells, the invasive properties, and the proliferation. And even, well, we lost a little bit of the title there, but the temozolazide, you can actually enhance the benefit of that through these microRNAs. Liquid biopsy. Again, these exosomes are be specific for meningiomas, different types of meningiomas. So theoretically, or very likely in the next few years, we'll be doing liquid biopsies, taking blood from a patient, say after surgery for meningioma, to detect recurrences. We won't have to do as many scans. Inflammation from ischemic stroke, subarachnoid hemorrhage, and brain injury. There's some commonalities in some of the mRNAs that are secreted in these conditions. And so that leads to, there may be some common treatment modalities as well. Cerebral vasospasm, a common problem for those who are cerebrovascular surgeons. It's very likely down the road we'll have this microRNA 148B-3P may be a therapeutic mechanism to decrease the vasospasm. We're seeing the microRNA 27A, this is in an animal model, but quite beneficial as far as promoting recovery after ischemic stroke. Just to go back and show you, this is a mesenchymal stem cell, secretes an exosome with these various microRNAs and other proteins that encourage angiogenesis in, say, post-ischemic injury. Saliva microRNA signature in children with TBI. You can see the heat map there, the various microRNAs. In the green on the right, there's 14 patients, and in the 9 on the left, the red, are controls. Quite a different heat map of the mRNAs, and so these can serve as a marker for predicting how children will do after traumatic brain injury. I mentioned this paper. This was funded by various agencies in Germany. Almost 3,000 participants looking at microRNAs in cognitive domains, and they found a couple of these microRNAs were associated with cognition and cortical thickness. These patients had MRI scans. And so some of these may actually be mechanisms for inhibiting the degeneration or the neurodegeneration in cognitive decline. Alzheimer's disease, a similar study, over 600 patients and looking at over 500 microRNAs. 15 of these showed evidence that they're involved in the Alzheimer's disease process and may serve as a therapeutic modality. Finally, with regard to this end of things, in severe depression, this microRNA 451A is correlated to lower levels in people who are severely depressed. You can have an animal or a rodent model of chronic restraint stress, which is a form of depression in rodents. And they found the same thing, that this was underexpressed. And so providing more microRNA 451A may prove to be a treatment for depression. And this is just showing some of the benefits in this animal model by enhancing this 451A. Well, a lot of your spine surgeons, disc degeneration, that's microRNAs are involved in that as well. 155-5P is a pro-inflammatory mediator, and so by inhibiting that, it may be a treatment down the road for low back pain and disc degeneration. And this is just showing on the left, if you enhance the 155-5P, you see various benefits, and on the right, if you inhibit it, it has a negative effect. Another paper, again, these are all from 2024. This is a very rapidly evolving field. One of the problems, if you're going to treat with these microRNAs, is how do you get them to hang around or go to the right place? And these nanoparticles are a way of allowing to increase the targeting capabilities. This 150-5P, remember, the one previous study is 155-5P, so it's probably pretty closely related structurally. And again, these can show efficacy for lumbar disc disease. And this is just showing the model they used in animals. In the upper left, you see they induced a disc disease in an animal, took the adjacent normal disc as a control. And you can see the heat map in the upper right, really quite different. And that 150-5P is one of those microRNAs, which is very different expression in a normal disc versus an abnormal disc. And at the bottom, you see the histology. And the bottom level is the histology for the one that was treated, significantly improved over the wild type or control disc disease. And finally, one more example of another microRNA involved in disc degeneration. Again, these are being used in animal models for the most part, although some of this has been documented in human disc disease in vitro. Ligamentum hypertrophy, another lumbar stenosis is a big problem for a lot of spine surgeons. And you can see here, there's a decrease in the collagen to, elastin to collagen ratio in hypertrophy ligamentum flavum. These are samples were taken from humans and tested in vitro, but also in an animal model, as you see here. And they actually showed that you can, in this rodent model, can decrease the ligamentum flavum hypertrophy with these two microRNAs, 146A and 2221. So again, I think very promising field for lumbar surgery. We may be injecting microRNAs instead of cutting. So fractures, osteoporotic fractures, and bone mineral density are also affected by microRNAs. I'll just finish with one example of this mesenchymal stem cell-directed nanovesicle. Macrophages know where to go. Stem cells don't know where to go. So these group infused the membranes of a macrophage with the content they want to secrete in spinal cord injury. And you can see here how they, whoop, I guess that's it. So anyway, next speaker.